There is a creature living in the shallow, sun-warmed waters of the Indo-Pacific that appears, at first glance, to be doing absolutely nothing. It sits on the sandy bottom, or nestles among coral rubble, or tucks itself just beneath the surface of the sediment, with only the tip of its shell visible. It moves so slowly that it is easy to mistake it for a decorative stone, for a discarded shell, for something entirely without menace. Tourists spot them on snorkeling trips and reach down to pick them up, attracted by the beautiful geometric patterns etched across the surface, the neat mosaic of brown and white and cream, the satisfying weight of the thing in the palm of a hand. Some of those tourists have died within hours of doing so. The cone snail is one of the most venomous animals on the planet, and the fact that it looks like a collectible trinket is precisely what makes it so dangerous to the people who do not know what they are holding. But the human danger, as extraordinary as it is almost beside the point when you start to understand what this animal is truly capable of. The real story of the cone snail is not about accidental human encounters at all. It is about one of the most sophisticated, most precisely engineered, and most spectacularly effective predatory systems that evolution has ever produced. A slow-moving mollusk, an animal without limbs, without speed, without the powerful musculature of a predator, has developed a method of killing fast-moving fish, almost before those fish have had time to register that anything has happened. If you find that genuinely astonishing, if you want to understand how a snail can outmaneuver a fish, then stay with this. Subscribe so you never miss a story like it, because there are far more of these stories in the ocean than most people have ever been told. To understand what makes the cone snail so remarkable, it helps to begin with the problem it has to solve. The cone snail is a mollusk, which places it in a broad category of animals that includes slugs, clams, and octopuses. Like most snails, it has a muscular foot that it uses to move across the substrate in a slow, gliding motion. It cannot chase anything. It cannot run down prey. It cannot leap, strike, or rush. It is, by the standards of the ocean, glacially slow. And yet the worm-hunting species have managed perfectly well for millions of years by pursuing worms that are equally slow and equally easy to catch. Those worms do not require any special weaponry. But at some point in the evolutionary history of the cone snails, a subset of the group made a remarkable dietary decision. They began hunting fish. Think about what this means. Fish are among the most agile animals in the ocean. They have evolved over hundreds of millions of years to detect predators, to respond in fractions of a second, to accelerate from standstill to full speed in a movement so fast that the human eye struggles to track it. The escape reflex of a fish, triggered by a pair of giant neurons in the brainstem called the Mouthner cells, is one of the fastest reflexes in the vertebrate kingdom. The moment a fish detects a threat, its body curves, its tail powers through the water, and it is gone before the predator has completed its strike. Eels, barracuda, moray, grouper, all of these fast and capable predators catch fish through speed, through ambush, through the relentless refinement of their own attack systems over millions of years. The idea that a snail might join this group seems laughable. And yet the fish-hunting cone snails have not simply joined this group. In terms of the speed and efficiency with which they immobilize prey, they may be unmatched by any predator alive. The solution the cone snail evolved is not a better body, it is a better weapon. And the weapon it built is, in the strictest engineering sense, a harpoon. Deep inside the body of a fish-hunting cone snail, behind the head, lies an organ called the venom bulb. Connected to this bulb is a long, coiled tube, the venom duct, which leads forward to a hollow, disposable, needle-sharp tooth called the radula tooth. In most snails, the radula is a rough, ribbon-like structure used for scraping food. In the cone snails, it has been modified beyond recognition, converted into a single, barbed, hypodermic weapon. When the cone snail is ready to strike, this tooth is loaded into the tip of a flexible, muscular tube called the proboscis. The proboscis can be extended in almost any direction from the snail's body, and it can swing around to reach a target behind or to the side of the snail's shell, not just in front of it. This is not a passive defense mechanism. This is an active, directional, multi-angle delivery system for one of the most complex venoms on Earth. When the proboscis makes contact with a fish, the venom bulb contracts. Pressure builds in the venom duct. The radula tooth fires forward through the proboscis tip with extraordinary force, punching through the fish's scales, anchoring in the flesh with its backward-facing barbs, and simultaneously injecting venom into the wound. The entire strike takes a fraction of a second. The fish is harpooned before it can escape, and then the venom takes over. The cone snail venoms are among the most complex biochemical substances produced by any animal. They are not simple proteins. They are not a single toxin delivered in quantity. They are intricate cocktails, mixtures of dozens or sometimes hundreds of individual compounds, each with a specific target in the nervous system or the musculature of the prey. Scientists call these compounds conotoxins, and the sheer variety of them is extraordinary. Different cone snail species produce different combinations of conotoxins, and within a single species the venom can change depending on whether the snail is hunting or defending itself. The same animal produces different chemical weapons for different situations, which is a level of pharmacological sophistication that researchers are still working to fully understand. The fish-hunting species have developed a particular strategy that reveals just how precisely these venoms have been refined. When a piscivorous, meaning fish-eating, cone snail is preparing to strike, it does not simply jab the fish and hope for the best. Many species release a cloud of specialized compounds into the water around the fish before the physical strike even happens. These compounds begin to act through the fish's gills and skin, crossing into the bloodstream, reaching the nervous system. By the time the harpoon fires, the fish is already beginning to be affected. The venom delivered by the strike then completes the process with remarkable speed. Different conotoxins block different channels. Some block the sodium channels that allow nerve impulses to travel, silencing the signals that would trigger the fish's escape response. Others block the potassium channels that regulate the recovery of nerve cells between firings, preventing the nervous system from resetting itself. Still others act directly on the muscle, preventing contraction regardless of what the nerve signals are saying. The fish is paralyzed not by a single mechanism, but by a coordinated assault on multiple systems simultaneously. The result, from the fish's perspective, is an almost instantaneous loss of the ability to move. The Mouthner cell reflex, that fastest of all vertebrate escape mechanism, does not fire, or if it fires, the signal goes nowhere. The muscles do not respond. The fish hangs in the water, motionless, and the slow cone snail moves in to engulf it whole. A predator that cannot chase its prey has solved the problem by making it irrelevant. The prey does not escape because the prey cannot move. The speed of the predator is rendered entirely beside the point. This is the genius of the cone snail. Not the harpoon itself, which is impressive enough, not the venom alone, which is extraordinary on its own terms. But the combination of the two, and the way in which the venom has been so precisely tuned to the nervous and muscular systems of fish that it can shut down the escape response almost before the fish knows what has happened. Evolution has solved, over millions of years of refinement, a problem that looks impossible. A slow animal has become an effective fish killer by changing the rules of the encounter entirely. There are roughly 700 species of cone snail currently known to science, and they inhabit the tropical and subtropical oceans of the world, with particular richness in the Indo-Pacific region. They range from the small and relatively unassuming species that hunt polychaete worms in the sand, to the large and extravagantly beautiful species that hunt fish on coral reefs. The worm-hunting species are the most numerous and the most widespread, and they are what most people encounter when they walk along a tropical beach or snorkel over a reef flat. Their venom is present but generally not powerful enough to seriously harm a healthy adult human. The mollusk-hunting species are intermediate, pursuing other snails and small bivalves with a venom calibrated accordingly. But the fish-hunting species, perhaps 30 or so out of the 700, are the ones that carry venoms capable of killing a person, and they are the ones that carry the full complement of the cone snail's most remarkable innovations. The geography of cone snail diversity is closely tied to coral reef systems. The Coral Triangle, that region of the Pacific bounded by Indonesia, the Philippines, and Papua New Guinea, is the global center of cone snail diversity, just as it is the center of coral reef diversity generally. Here in warm, shallow, reef-associated waters, with the extraordinary productivity and structural complexity of the reef providing habitat and prey, the cone snails have radiated into their greatest variety of forms. Some species have adapted to the deeper reef slopes, hunting in 20 or 30 meters of water. Others work the sandy flats between reef patches, buried beneath the surface during the day and emerging to hunt at night. Still others haunt the rubble zones at the base of reef structures, where broken coral provides cover and where small fish shelter during the hours of darkness. This nocturnal behavior is characteristic of the fish hunting species in particular. During the day, a cone snail that hunts fish is largely inactive, tucked away beneath sand or rubble, protected by its shell and by the toxicity of its venom. At dusk, it becomes active, extending its siphon above the substrate to sample the chemical signals in the water. Fish release compounds as they breathe, as they feed, as they move. The cone snail's olfactory system, centered on a structure called the osphradium, is exquisitely sensitive to these chemical traces. It can detect a fish in its vicinity through the water chemistry alone, building up a picture of the environment entirely through chemistry, in the way that a hunting mammal builds up a picture through scent. When the cone snail detects a fish nearby, its behavior changes. The proboscis begins to extend from the head, probing the water. The animal moves toward the source of the chemical signal, following the gradient of concentration, in the way that a predator follows a scent trail across the ground. This movement is slow, by any standard, but in the darkness, with the fish resting or feeding in a crevice, slow is enough. The cone snail does not need to be fast in this phase of the hunt. It needs only to get close enough for the proboscis to reach. Some fish hunting cone snails have evolved an additional behavioral refinement that makes the closing phase of the hunt even more effective. They extend the proboscis to its full length and hold it motionless, waiting for the fish to move close enough on its own. This is ambush predation, the strategy that has been independently evolved by so many of the ocean's most effective predators, from the scorpion fish to the frogfish, from the wobbegong shark to the mantis shrimp. The cone snail sits in the dark, chemically alert, proboscis extended, and waits. The fish moves within range. The harpoon fires. The encounter is over. The cone, known as Conus geographus, is perhaps the most notorious species, and it illustrates the full range of the cone snail's capabilities with particular clarity. It is a large species, reaching lengths of 15 centimeters or more, and it has a beautifully patterned shell of white and brown arranged in irregular geographic patterns, which is precisely how it earned its common name, the geography cone. It is also among the most venomous animals in the ocean, and it is responsible for the majority of recorded human deaths from cone snail stings. It hunts fish at night on Indo-Pacific coral reefs, and its venom is calibrated to shut down the nervous systems of teleost fish with extraordinary speed. The same venom, when delivered accidentally to a human, can cause progressive paralysis, respiratory failure, and cardiovascular collapse. There is no anti-venom. Treatment is entirely supportive, meaning that medical staff can only try to keep the victim breathing and their heart beating while the venom runs its course. Death can occur within hours of the sting. What makes Conus geographus particularly dangerous to humans is not just the potency of its venom, but the manner in which it delivers it. The proboscis of this species is unusually long and flexible, capable of extending around and under the shell to reach virtually any direction. A person who picks up a geography cone by the shell is not necessarily safe. The proboscis can reach around the shell to sting the fingers holding it. The barbed radular tooth, once fired, is held in the flesh by its backward-pointing barbs, and the venom is injected with each pulsation of the venom bulb, which can continue even after the initial strike. The sting is often described as feeling no worse than a mild bee sting at first, which is precisely what makes it so insidious. There is no immediate dramatic pain to warn the victim that something serious has happened. The venom is already doing its work before the victim has fully registered that they have been stung. The specific toxins in Conus geographus venom include a compound that has become famous in a very different context, the compound known as conotoxin Mr. Via, which belongs to a group of conotoxins that target voltage-gated calcium channels, and another compound so potent at blocking neuromuscular transmission that even tiny quantities cause complete paralysis of the muscles involved in breathing. But among the most remarkable compounds in this and other piscivorous cone snail venoms is a family of molecules called conconitsins, and a smaller set of compounds including what researchers have described as lightning strike cabal venom, a specialized mixture that acts so rapidly on the fish's nervous system that the escape reflex is completely abolished within fractions of a second of the venom entering the bloodstream. The fish-paralyzing cocktail that Conus geographus and related species use begins, as described earlier, with a pre-strike chemical cloud. This has been studied in the laboratory by researchers who observed that fish in the vicinity of a hunting cone snail begin to show signs of neurological impairment before any physical contact has occurred. They lose coordination, their movements become irregular, their ability to orient and respond to stimuli is impaired. By the time the harpoon arrives, the fish is already compromised. The strike then delivers a further, concentrated dose of different compounds, targeting the specific channels and receptors that the cloud has already begun to affect. The two-phase system, cloud followed by strike, ensures that even a fish with extremely fast reflexes has no opportunity to escape. The evolution of this system raises profound questions about how it developed in the first place. The ancestral cone snails were worm hunters, and the transition from hunting slow worms to hunting fast fish represents a shift in prey that is enormous by any standard. Researchers believe the transition was gradual, progressing through stages in which increasingly mobile prey were targeted with increasingly refined venoms. Some species alive today hunt both worms and small fish opportunistically, and these may represent an intermediate stage in the evolutionary progression. The venom of these generalist species is less precisely tuned than that of the dedicated fish hunters, containing a broader mix of compounds that are effective against a wider range of prey. Over time, in lineages that increasingly specialized on fish, the venom composition shifted toward the precise, multi-target, multi-phase system that the dedicated piscivorous species use today. The radular tooth itself has also been refined over evolutionary time. In worm-hunting species, it is smaller and simpler. In fish-hunting species, it is longer, more strongly barbed, and capable of penetrating the much tougher tissue of fish skin and scales. The venom bulb is correspondingly larger and more powerful, capable of generating the pressure needed to inject venom rapidly and in sufficient quantity to overwhelm the fish's nervous system before the escape reflex completes. Each component of the system has been co-refined with the others, which is a beautiful example of what evolutionary biologists call integrated evolution, in which multiple traits evolve together because they are all part of a single functional system. The shells of cone snails are among the most prized objects in the history of shell collecting, and this has had profound consequences for the animals themselves. Before the era of synthetic materials and mass production, rare shells commanded extraordinary prices. The geography cone, along with several related species, was sought by collectors across the world, and individual specimens of particularly fine quality have sold for prices equivalent to thousands of modern dollars. The trade drove collection pressure across the Indo-Pacific for centuries, and while the cone snails are not currently considered endangered as a group, local populations in heavily collected areas have been significantly reduced. The irony of the geography cone's story is that an animal which is genuinely dangerous to handle was nonetheless collected in enormous quantities by people willing to accept the risk for the beauty of the shell. Today, the cone snails face a different set of pressures. Coral reef degradation reduces the habitat quality and structural complexity that the fish-hunting species in particular depend on. The fish on which they prey are themselves heavily exploited in many parts of the Indo-Pacific, reducing the availability of prey in areas where fishing pressure is high. Sedimentation from coastal development smothers the sandy and rubble habitats where cone snails forage and shelter. Climate change is altering the temperature and chemistry of reef systems in ways that affect both the cone snails themselves and the ecosystems on which they depend. None of these pressures is unique to the cone snails, but together they represent a significant threat to animals that, despite their extraordinary weaponry, are slow to reproduce and highly dependent on the integrity of reef ecosystems. But the story of the cone snail in the modern era is not only a story of threat, it is also, and perhaps most remarkably, a story of one of the most unexpected and important medical discoveries of recent decades. The conotoxins, those hundreds of precisely targeted neurological compounds that the cone snails use to paralyze their prey, have turned out to be among the most useful pharmacological tools that science has ever encountered. The reason is precisely the quality that makes them so effective as venom, their specificity. Each conotoxin targets a specific type of ion channel or receptor with extraordinary precision, ignoring all others. This specificity is exactly what pharmaceutical chemists spend years trying to achieve when developing new drugs, and the cone snails have achieved it through evolution over millions of years. The most famous example is a drug derived from the venom of the cone snail known as Conus magus, the magician's cone, a smaller species than the geography cone, but nonetheless a fish hunter with a potent venom. One of the conotoxins in its venom, a compound known as omega-conotoxin MVA, was found to block a specific type of calcium channel in pain-sensing neurons with extraordinary precision. When this compound, or a synthetic version of it, is injected into the fluid surrounding the spinal cord, it blocks the transmission of pain signals, through pathways that conventional pain medications do not reach effectively. The resulting drug, ziconotide, sold under the trade name Prialt, was approved for medical use in the United States in 2004. It is now used to treat severe chronic pain in patients who have not responded to other treatments, including patients with cancer, spinal cord injury, and other conditions causing intractable pain. It is, in the most literal sense, a venom-derived drug. A compound designed by evolution to paralyze fish is now relieving suffering in human patients for whom no other treatment was effective. This is not an isolated case. Researchers around the world are studying the venoms of hundreds of cone snail species, identifying conotoxins, and testing them against a wide range of molecular targets. Some are being investigated as potential treatments for epilepsy because of their ability to modulate the ion channels whose dysregulation underlies seizure activity. Others are being studied for their potential in treating cardiovascular disease because of their effects on the channels involved in regulating heart rhythm. Still others are being explored as tools for mapping the nervous system because their precise target specificity makes them invaluable for blocking specific pathways without affecting others, allowing neuroscientists to dissect the circuitry of the brain and spinal cord with a resolution that other tools cannot achieve. The sheer number of conotoxins available for study is staggering. It is estimated that the 700 or so species of cone snail produce, between them, somewhere between 50,000 and 100,000 distinct conotoxin compounds. As of the time of writing, only a small fraction of these compounds have been isolated, characterized, and tested. The vast majority of cone snail species have never had their venoms systematically analyzed. This means that hidden within the shells of animals sitting on coral reef rubble across the Indo-Pacific, there is an enormous library of precisely engineered neurological compounds, most of them unknown to science, each of them representing a potential tool for medicine or neuroscience that evolution has already done the hard work of designing. The destruction of reef ecosystems and the reduction of cone snail populations is not only an ecological loss, it is the potential loss of a pharmacological resource of incalculable value. The discovery of the cone snail's medical potential has changed the way researchers and conservationists think about venomous animals generally, and it has given a new urgency to the work of understanding ocean biodiversity. If a slow-moving snail can produce compounds that cure intractable pain, what might other reef invertebrates contain? The ocean is full of animals that produce complex chemical compounds, many of them for defensive or predatory purposes, and many of those compounds have never been studied. The cone snail story is a vivid illustration of the principle that the most unexpected organisms can harbor the most valuable discoveries, and that losing biodiversity before we have had the chance to study it is a loss that cannot be recovered. To watch a fish-hunting cone snail in action, in the darkness of a tropical night on a coral reef, is to watch one of the most quietly spectacular events in the natural world. The snail is barely moving, the proboscis extended, the siphon probing the water. A small goby, perhaps 5 centimeters long, rests in a crevice in the coral rubble a few centimeters away. It is not alarmed. It has no reason to be alarmed. There is no fast-moving shadow, no sudden rush of water, no pressure wave of an approaching predator. There is only the slow presence of the cone snail and the chemical cloud that has already begun to spread through the water of the crevice, already beginning to reach the fish through its gills. The proboscis moves toward the fish, slowly, almost imperceptibly. It makes contact. The harpoon fires. The goby gives a single, brief convulsion, the only movement it will make, and then it is motionless. The cone snail begins to engulf it, the mouth expanding far beyond what the shell's aperture suggests should be possible, the fish disappearing slowly into the animal's body. By the time the first pale light of dawn begins to filter down through the water, the cone snail is buried again beneath the sand, the goby gone, the only evidence of what has happened a small hollow in the substrate where the snail rested. The reef resumes its appearance of tranquility. The cone snail is, by any measure of behavioral or biochemical complexity, a deeply sophisticated predator. But it is also an animal that challenges something more fundamental than our understanding of hunting strategies. It challenges our intuitions about what a predator can look like. We expect predators to be fast, muscular, visually arresting. We expect them to announce their nature through their form. The cone snail announces nothing. It looks like a shell on the sand. And it is precisely this invisibility, this profound quietness of presence, that makes it one of evolution's most elegant solutions to the ancient problem of how to catch something that would otherwise escape. There is a broader lesson here about the way life works in the ocean, and perhaps more generally, about the way evolution works. The most impressive adaptations are not always the most obvious ones. The fastest fish are not necessarily the most dangerous predators. The most venomous animals are not the ones that look most threatening. The ocean is full of creatures that have solved the problem of survival through strategies that are invisible, or slow, or chemical, or behavioral, rather than through raw physical power. The cone snail is an extreme example of this principle, but it is not an isolated one. Consider the mantis shrimp, which delivers a strike to its prey so fast that the water around it briefly cavitates, the bubbles collapsing with a force that can stun or kill even if the strike itself misses. Consider the pistol shrimp, which snaps its claw shut so quickly that a jet of cavitation bubbles produces a shockwave powerful enough to stun small fish. Consider the electric ray, which generates a burst of electricity sufficient to knock down a person, not to defend itself in the moment of attack, but to stun prey at a distance before it can react. Each of these animals has evolved, as the cone snail has, a method of overcoming the reflexes and the speed of fast-moving prey without matching those reflexes or that speed directly. Each of them has found a way to make the prey's speed irrelevant by changing the nature of the attack. The cone snail did this with chemistry. It built over millions of years of evolutionary refinement, a venom so precisely targeted at the nervous systems of fish that the fish's speed, its reflexes, its Mothner cell escape response, its millions of years of coevolution with fast-striking predators, all of it counts for nothing. The venom arrives before the reflex can fire. The paralysis is complete before the fish has registered the threat. The hunt is over before it has properly begun. In the waters of the Indo-Pacific, on the reef flats of the Coral Triangle, along the sandy margins of island coastlines from Japan to Australia, the cone snails are hunting tonight, as they have been hunting for 50 million years or more. They are buried in the sand, probing the water with their siphons, extending their proboscises toward fish resting in the dark. In laboratories in California, in Germany, in Australia, researchers are studying the compounds in those venoms, identifying new molecules, testing new drugs. In hospitals and patients with no other options, some of those molecules are already at work, blocking pain signals in pathways that nothing else can reach. The slow predator that looks like a decorative shell is rewriting what we know about venom, about evolution, about the nervous system, and about the origins of medicine. It asks us to look more carefully at the things that seem unremarkable, at the animals that sit still, at the biochemical complexity hidden in a shell on the sand. It asks us to consider that the most powerful strategies are not always the most visible ones, that speed is not the only way to catch something fast, that a weapon built of chemistry and patience can outperform a weapon built of muscle and speed. The cone snail has been doing this for longer than our species has existed. It will, in all likelihood, continue doing so long after we have come and gone, unless we are careless enough with the reefs and the oceans and the diversity of life they contain to foreclose that future before it arrives. That would be a loss not only for the reefs, and not only for the fish, and not only for the science, but for the possibility of understanding, which depends on there being something left to understand. The cone snail, hidden in its shell, firing its invisible harpoon in the dark, is one of the ocean's deepest arguments for why that understanding matters. The diversity of cone snail species is itself a story worth following into greater depth, because the variety of forms and strategies within this single family of mollusks is extraordinary. As already noted, the worm-hunting species represent the majority of the roughly 700 known species, and they inhabit a wide range of marine environments, from intertidal rock pools to relatively deep offshore sediments. These species are generally smaller, with narrower shells, and their venoms are less complex and less dangerous than those of the fish hunters, though they are by no means harmless. The worm-hunting cone snails track their prey through the chemical traces that polychaete worms leave as they move through the sediment, following the mucus trails with a precision that speaks to the same highly developed chemosensory system that the fish hunters use to locate their prey. When a worm-hunting cone snail catches up with its prey, the strike is the same. The radular tooth fires, the venom immobilizes the worm, and the snail moves in to consume it. The mollusk-hunting species occupy an interesting intermediate position in terms of both diet and venom complexity. They pursue other gastropods, including other cone snails, as well as bivalves, and their venoms include compounds that are effective at disrupting the neural control of the mollusk foot, preventing the prey from withdrawing into its shell or from clamping shut its valves. Some mollusk hunting species have been observed eating closely related cone snail species, which raises interesting questions about predator-prey dynamics within the family itself, and about whether the venoms of the prey species represent any form of defense against predation by their relatives. Among the fish-hunting species, there is further variation in strategy and in the composition of the venom cocktails used. Some species are active hunters that move across the reef at night, following chemical signals to locate sleeping fish. Others are more ambush-oriented, remaining buried or concealed, and extending the proboscis to intercept fish that move close enough to the snail's position. The size of the prey taken by different species varies considerably. The larger fish-hunting species, such as Conus geographus and Conus tulipa, can take prey of quite substantial size relative to the snail itself, with the elastic body of the snail capable of expanding to accommodate fish considerably larger than the shell's aperture would suggest. The venom delivered in these strikes is correspondingly large in quantity, which is why the stings of these species are so dangerous to humans, who are inadvertently harpooned. Conus tulipa, commonly called the tulip cone, is another species worth examining in some detail, because it illustrates a particular refinement of the fish hunting strategy. This species has been observed using what researchers have described as net hunting behavior, in which the snail spreads its propodium, the front portion of the muscular foot, out across the substrate in a broad, flat sheet. Fish resting near the snail on the bottom are partially enclosed by this spread tissue before the harpoon fires. The spreading of the propodium is thought to serve multiple functions. It may help to trap the fish against the substrate, limiting the initial escape movement even before the venom takes effect, and it may help to position the proboscis for an accurate strike. It is a behavioral refinement layered on top of the biochemical refinement of the venom, adding a physical dimension to a strategy that is already extraordinarily effective on the chemical level alone. The behavior of cone snails in response to threat is equally interesting. When disturbed, some species retract entirely into the shell, relying on the protection it provides. Others, however, respond to disturbance by extending the proboscis and attempting to sting whatever is disturbing them, which is the source of most accidental human stings. The defensive venom delivered in these circumstances is, in some species, different in composition from the predatory venom, containing a higher proportion of compounds targeting pain receptors and producing an immediate, sharp sensation designed to deter a predator rather than to paralyze prey. This distinction between predatory and defensive venom in the same animal is a remarkable pharmacological capability, requiring the snail to selectively deploy different mixes of compounds from its venom duct, depending on the nature of the stimulus it is responding to. The mechanisms by which this selection is made are not fully understood and represent an active area of research. The reproductive biology of cone snails is less dramatic than their predatory behavior, but no less interesting from an ecological perspective. Cone snails are broadcast spawners that release fertilized eggs in leathery capsules attached to the substrate. Each capsule contains hundreds to thousands of eggs, which develop through a larval stage called a veliger, a free-swimming larva that is part of the plankton for a period of days to weeks before settling on the reef and beginning to develop its shell. This planktonic dispersal phase means that cone snail populations on different reefs can be connected over considerable distances, with larvae carried by currents from one reef system to another. It also means that populations are vulnerable to disruption during this sensitive early phase, when larvae are exposed to the full range of planktonic predators and to the physical conditions of the open water. Growth rates in cone snails are relatively slow, with individuals of the larger species taking several years to reach reproductive maturity. Lifespans of 5 to 10 years or more are typical for the larger species, though precise longevity data are difficult to obtain for wild populations. The shell grows throughout the animal's life, added to at the aperture as the snail's body grows, with the inner whorls of the shell representing earlier growth stages preserved in the spiral architecture of the shell. The external patterning of the shell, which is produced by specialized cells in the mantle tissue, is added progressively as the shell grows, and the geometric complexity of cone snail shell patterns has attracted the attention of mathematicians and computer scientists interested in self-organizing systems. The patterns of some cone snail species, most notably Conus textile, can be described by simple cellular automaton rules, meaning that the pattern is generated locally, each cell in the mantle making a coloring decision based on the state of its neighbors, producing a global pattern of remarkable complexity from local rules of great simplicity. This has been recognized as an example of emergence, the production of complex patterns from simple rules, and it has influenced thinking in computational biology and complex systems science. The reef habitat on which cone snails depend is itself a system of extraordinary complexity and productivity, and the role of cone snails within that system is worth considering. As predators, the fish-hunting cone snails exert a selective pressure on the fish populations of the reef, targeting in particular the species that rest on or near the bottom at night, including gobies, blennies, and small wrasses. The fish that survive these encounters are those that, by chance, were not in the cone snail's path, or were alerted by some chemical signal early enough to move away before the snail approached closely enough to strike. Over many generations, there may be selection for behavioral traits that reduce vulnerability to cone snail predation, such as sheltering in crevices inaccessible to the snail's proboscis, or remaining more vigilant during the hours when cone snails are active. As prey, cone snails are eaten by a variety of reef predators, including cone snail-eating fish, such as triggerfish and pufferfish, which are able to pick up and manipulate the shell to get at the soft tissue inside while avoiding the proboscis, and by other mollusks, including the famous Triton's trumpet shell, Charonia tritonus, which is also a predator of the crown-of-thorns starfish, mentioned elsewhere in discussions of reef ecology. The Triton's trumpet is one of the few mollusk predators large enough and sufficiently well protected to tackle cone snails of moderate size, and it appears to be immune or at least highly resistant to the cone snail's venom. The mechanisms of this resistance are not fully characterized, but they represent another example of an evolutionary arms race played out in the chemistry of the reef. The question of how the fish-hunting cone snail lineages originally evolved from worm-hunting ancestors is one of the most interesting questions in molluscan evolutionary biology, and it has been approached through both molecular and morphological analysis. Molecular phylogenetics, the reconstruction of evolutionary relationships from DNA sequence data, has shown that fish-hunting species have evolved multiple times independently within the cone snail family, meaning that the radical shift to hunting fast, mobile prey is not the product of a single evolutionary event, but has been arrived at separately by different lineages on different occasions. This suggests that the evolutionary pressure to exploit fish as prey was strong enough in environments where fish were abundant and other predatory niches were available to drive the evolution of the necessary adaptations repeatedly. Each time, the evolution of a more powerful and more specifically targeted venom system preceded or co-evolved with the behavioral and morphological changes needed to deliver that venom effectively. This convergent evolution of fish hunting in multiple cone snail lineages is a reminder of the broader principle that when environmental conditions favor a particular strategy, evolution tends to arrive at similar solutions independently. The cone snails are a molluscan family that has found, multiple times, that the ocean is full of fast, nutritious fish, and that the way to catch them is not to become faster, but to become more chemically sophisticated. The same principle, in different forms, explains the convergent evolution of venom systems in snakes, spiders, jellyfish, and the other groups of animals that have independently discovered that chemistry can do what speed cannot. In the years since the discovery of ziconotide, the cone snail-derived pain medication, the pharmaceutical interest in cone snail venoms has grown considerably. Research programs at universities and pharmaceutical companies across the world are pursuing the identification and characterization of conotoxins for a wide range of medical applications. The compounds being investigated include candidates for treating conditions as diverse as Alzheimer's disease, schizophrenia, chronic inflammatory pain, post-surgical pain, cardiovascular arrhythmias, and the neuropathic pain associated with conditions like diabetes and multiple sclerosis. The research proceeds slowly, because the process of moving from an identified compound to a tested, approved drug is measured in decades rather than years, but the breadth of potential applications reflects the biochemical richness of the resource. One of the technical challenges in this research is obtaining the venom compounds in sufficient quantity for testing. Cone snails are not easy to maintain in captivity, and milking them for venom, the process used routinely for snake venom research, is technically demanding and potentially dangerous. Much of the early research relied on dissecting the venom ducts from specimens collected from the wild and analyzing their contents chemically. This approach is limited by the supply of specimens and by the ethical and practical constraints on collecting rare and ecologically important animals. Modern molecular biology has partially circumvented this problem by identifying the genes that code for conotoxin proteins and expressing those genes in bacterial or cell culture systems to produce the compounds in quantity. This approach is not perfect because the post-translational modifications that cone snail cells perform on the conotoxin proteins are often important for their biological activity and are not always replicated in bacterial expression systems, but it has substantially accelerated the pace of research. The broader point is that the cone snail, this quiet, slow, beautiful, and extraordinarily dangerous animal, occupies a unique position at the intersection of ecology, evolutionary biology, neuroscience, and medicine. Understanding it fully requires expertise from all of these fields simultaneously. The evolutionary history of its venom system is a story about arms races, about the refinement of targeting systems over millions of years, about convergent evolution and the repeated discovery of chemical solutions to physical problems. The ecology of its interactions with fish, with other predators, with the reef system as a whole, is a story about the complexity of food webs and the ways in which even a slow and apparently unimpressive predator can shape the community around it. The neuroscience of its venom is a story about the fundamental architecture of the nervous system and the ways,